Hello listeners, Yamina here. Welcome to the Dr. GPCR podcast. We are excited to share that the Dr. GPCR Ecosystem 2.0 platform opens to all beta testers starting June 15th, 2022. We plan on opening the ecosystem slowly to resolve any kind of kinks as soon as possible. You can visit us at ecosystem.drgpcr.com and explore the new site. Access is restricted to members of the GPCR field, and each sign-up will be approved based on one's involvement in the field. Once your sign-up is approved, you'll be able to enter the ecosystem. You'll also have the option to select a plan and get access to all things Dr. GPCR, including the upcoming Dr. GPCR Summit, access to new podcast episodes before they even get released to the general public, our new group discussion and forums, our new and improved job board, and Learning Center, where you'll be able to take a course or even prepare and share a course with your colleagues. Discover GPCR companies in one place and much more. Take advantage of everything that the new GPCR dedicated online playground has to offer today by visiting ecosystem.drgpcr.com and signing up. This is the only place where GPCR scientists, trainees, and GPCR organizations can thrive and where it's only about science and GPCRs. Think ResearchGate meets LinkedIn meets Amazon at the tip of your fingers. Visit ecosystem.drgpcr.com to start your journey today. Also, make sure that you mark your calendar for the third edition of the Dr. GPCR Summit. This year, the summit will be held between October 10th and 16th. Stay tuned for more soon. Visit ecosystem.drgpcr.com or visit our old website, which is still active, drgpcr.com, to explore everything that we've been doing in the past 2 years. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. And today I'm delighted to have on the Dr. GPCR podcast, Professor Graeme Milligan. Hi, Graeme.
Hi there. Nice to see you.
Nice to see you. So excited to have you on today.
Well, I'm looking forward to it as well. It's always nice to talk to people about things that excite you.
That's absolutely could not agree more. Okay, so let's start at the beginning. And if you would kindly give us a little introduction as to who you are and what do you do?
Okay, so as you heard earlier, my name's Graeme Milligan. I'm currently a professor of biochemistry at the University of Glasgow in Scotland, the United Kingdom. And I've been based in Glasgow for most of my professional life. In fact, I've been here now for more than 36 years.
Wow.
And working on GPCRs that entire time, as well as the time when I was a postdoc and indeed as a graduate student. So I've literally been in there since GPCRs weren't proteins and just were pharmacological concepts.
That is amazing. You know, I've worked in the GPCR field as I started very early on. The first time I heard about GPCRs was in an undergrad class, and the receptors and the concept and the pharmacology has followed me through the past 20 years. And sometimes I thought, okay, maybe I should have learned something else, but I don't regret spending so much time in the field. And it's good to hear that I'm not the only one who's so in love with.
Neither do I, but I guess like many people, when I was an undergraduate student, there were a number of themes around biochemistry that I was quite excited by.
Mm-hmm.
And when it came to trying to select what I might study for my PhD, I was quite torn between cellular signaling and bioenergetics.
Interesting.
And it's perhaps lucky or perhaps not that I ended up to go towards cellular signaling.
Well, I think the field is really grateful to have you on, and you've done. I remember as an undergrad and as a graduate student reading your papers, and I'm so excited that we're talking today. Let's talk a little bit about your story as a scientist. Let's go back to Graeme at 15 or maybe Graeme at 10. Did you always know that you wanted to be a scientist?
No, absolutely not. So I'm a little bit unusual in many ways because I was the first, and I think I'm still the only member of my family who's ever gone to university.
Okay.
I grew up on a dairy farm, and I think pretty much all the expectations was that I was going to milk cows for a living. My dad was quite keen on that. My mum less so, I think, based on the hours. Although as a scientist, I think I do almost as many hours as a farmer does in reality. It's just a rather easier life on the hands.
Absolutely.
So actually, it wasn't really until I was very close to the end of high school that the idea of biochemistry started to become an option.
Mhm.
I wasn't actually good enough at high school to get into university. So I went and took a job working in a local hospital where I worked in the biochemistry labs. So sort of doing blood gases and iron levels in blood and whatever. And I started going to night school.
Wow.
So I spent 2 years at night school, and then I decided that actually my day job was not very exciting and that I would try again to get into university and hope that the experience I had from doing something not immediately going to university after school might hold me in good stead. And
Wow.
luckily enough, I got accepted to do biochemistry at the University of Birmingham in the UK. So that's where my biochemistry came from.
That's amazing. But as a child, what were your interests? What were you interested in growing up on a dairy farm? How did you go from?
What I was mostly interested in was playing soccer, or obviously football to us.
Yes, football.
And
yes.
It seems like most of my early pictures seem to have me kicking a football around. And when I was a teenager, I actually played football semi-professionally.
Wow, some from semi-professional football player to biochemist. Phenomenal.
Yeah. And I think I quickly discovered that once I started to play with guys who were pretty good, that clearly I wasn't, and that was not going to be a successful career for me, no matter how much I might have liked it.
That's phenomenal. So you got into the biochemistry program, and what led you towards research? Because doing an undergrad in biochemistry doesn't necessarily mean research career.
Yeah, no, absolutely. But one of the surprising things when I went to university is because I'd not been successful at getting into university when I left high school, I was a little surprised at the end of first year that I came out pretty much top of the class.
Do you think
and that continued, luckily, through the 3 years of my undergraduate course. So it was almost as if I fell into thinking that further work in biochemistry would make sense. And I had a very influential teacher when I was at the University of Birmingham. His name is Bob Michel, and he was one of the founding figures of work on inositol phosphate signaling. So I think because of his influence, I wanted to do cell signaling in some description. But it turned out that the lab I went to for my PhD was actually starting to study and to understand the nature of G protein coupled receptors. And at that time, the only receptor or the only G protein that was known was GS, the one that couples to cyclic AMP stimulation. And my PhD was essentially trying to understand if there might be more members of the family.
Wow. Amazing.
Using very simplistic, of course. Now, of course, this was 40-something years ago. So, the techniques weren't quite where we are these days.
Absolutely. And it's funny because I when I talk to my podcast guests, we talk about the technology and how far we've come in the field. Now we do assays in 384 or even higher throughput. And 20 years ago, I was using radioactivity to quantify cyclic AMP accumulation in cells.
Sure, and I certainly was using radioligand binding assays. In fact, looking at what at that time people thought was the one and only member of the muscarinic acetylcholine receptor family. We didn't at that point know there was going to be 5 in humans, and really studying some quite simple aspects of whether that receptor was coupled to a G protein or not. So starting to use nucleotide analogs that could disrupt the interaction between a receptor and the G protein, the sort of guanine nucleotide shift that I still teach my students as when they're coming to my classes as undergraduates, and the sort of thing that, as you were saying earlier about Bob Michel, the idea of a guanine nucleotide shift encouraging those that worked on the beta receptor to recognize there must be something that needed GTP in the system to mediate function.
Amazing. You made a very interesting point, and I think it's important to put it out there, is the fact that you didn't get into university right away after high school, and still you persevered, and then when you got in, you realized that you were very quickly top of the class. And I think and this is especially for students and trainees, you need to find what you really like, and then it just, quote unquote, comes naturally.
Absolutely. I think you do need to find your niche in life, whether it's in science or in something else. I'm sure I could have been happy doing other things, but I very much doubt I would have been as happy as the career that this has allowed me to develop. And maybe for folks these days, even in the UK, it perhaps comes as a surprise now that almost half high school graduates go to university. In those days, it was less than 5%. And you got a grant from the government to go to university because they were training a group of people for higher level work. So the fact that I didn't get in when I left high school didn't mean I was completely useless at school. I just wasn't in the absolute upper echelons.
Yeah,
but then
absolutely.
I found something I loved. And I think if you do anything you love, you're willing to put in the hours and the time and take the rough with the smooth, as they say, when the experiments don't go well.
Yes, and the experiments tend not to go well 90% of the time.
Absolutely. My PhD was completely and utterly a failure, as far as I can see. We published very little and nothing of significance. And it was a miracle in some ways that when I applied to get a Fogarty International Visiting Fellowship at NIH, one of the labs there wrote back and said, please come.
Wow.
So I was very pleasantly surprised to be offered a postdoc position at NIH in Bethesda.
I was as well at NIH in Bethesda for a postdoc. Oh,
yeah, and I went there to work with a guy called Werner Klee, who worked on essentially opiate receptors. Mm-hmm. And we had a very small team where one person was trying to purify the receptor. I was trying to purify a corresponding G protein. And the third person in the team was trying to reconstitute them and put them back together.
How phenomenal. You mentioned that you first started working officially on GPCRs during your PhD. When did you first hear about GPCRs?
I guess during my undergraduate work, because although the focus of the cell signaling courses there were very much about lipid signaling, and there was a feeling that, the discovery of calcium mobilization and the mechanisms by which inositol phosphates were generated and regulated, that was really the focus of it. And although there was a recognition that something must be stimulated to turn it on, we had rather little about the nature of the receptor, largely because the nature of the receptor wasn't very well worked out or clear. even at that time, people were only starting to get to grips with rhodopsin as the prototypic GPCR.
Wow. And after your postdoc at NIH working on opioid receptors and purifying G protein, corresponding G proteins, where did you went back to the UK? How did you end up where you are today?
Yeah, so I did go back to the UK immediately after my postdoc at NIH. And what I was planning to do was to do a second postdoc with a guy called Miles Housley, who at that time was in Manchester in the UK. But by the time I'd applied to him and he said, "Oh, come over and talk to me," he'd taken a new job as professor in Glasgow. And when I arrived, he said, "Would you quite like a permanent job as the equivalent of assistant professor?"
Wow.
And I said, "Well, why not? I don't really have the experience for it, but if you trust me, I'll go for it." And as I said to you earlier, Yamina, 37 years later, I'm still here.
Amazing, amazing. And what did you start working on as an assistant professor?
Well, I was already, because I said to you that I was working on purification of G proteins, during the time I was at NIH, I made a very strong collaborative interaction with a guy called Alan Spiegel, who was head of one of the institutes at NIH, and his German postdoc called Peter Gierschik, who is now professor of pharmacology at the University of Ulm in Germany. And we started to recognize that by making antibodies against the transducin protein that's involved in the rhodopsin to cyclic GMP signaling cascade, we found that antibodies we'd made against rhodopsin, some of them, saw proteins in other tissues, which turned out to be the GI and GO family of GPCRs, of G proteins. So we published very extensively for a period of time between about 1985 and 1987. And that really set the framework for my own individual lab. So when I moved back to the UK, my first grants were all really on G proteins and their function rather than receptors. But I guess after a few years, I decided that it was going to be impossible to stay in that area without really starting to explore the receptors as well. And therefore, much of the effort in my team moved upstream to the nature of the receptors, how they signaled, how they interacted with G proteins and their cell activity.
Amazing.
And I guess quite early on, I'd started to develop technologies that would allow screening for ligands at receptors.
What were those? I'm sorry. What were those technologies? There was the lag.
The very first technology that I developed and patented was really a strategy in which we found a way in which you could destabilize a GPCR by mutating residues that were close to the so-called dry domain, so in intracellular loop 2, and that reduced the half-life of the protein. But we found that if you added an antagonist that bound to the receptor, it would reverse that destabilization. So if you grew cells with one of these destabilized receptors in the presence of a molecule that bound to the receptor, the amount of the receptor would go up. And therefore, if you linked it to, for example, a luciferase, or indeed a fluorescent protein, you could either measure enzymatically or by fluorescence the fact that a molecule from your screen must have bound your receptor of interest.
Amazing. You were really
and
ahead.
Particularly for orphan receptors, sorry to interrupt there, Yamina,
Yes.
but particularly for orphan receptors, you could then screen without any knowledge of what the ligands for the receptor was to be. And that turned out to be quite a successful strategy.
That's exactly what I wanted to say, is that I think you were really ahead of your time when you think about this, because it's a method that really allows you to screen independently of the nature of the receptor, whether you know it or not.
Indeed. And this was perhaps my first foray into what these days you might call translational pharmacology, because I licensed that technology to a small company that one of actually a former PhD student at the University of Glasgow had set up in Southern California.
Wow. What's the name?
It's called Arena Pharmaceuticals. Mm-hmm. And some folks might notice that just a couple of months ago, Arena Pharmaceuticals was bought by Pfizer for $6.7 billion. Wow.
That's
so
fun.
It's interesting how things develop. That did take 25 years, to be fair. And there were a few hiccups on the road. But I was part of Arena at the time when we had a door into an office, but no labs.
Wow.
And exciting times to get involved in the biotechnology of GPCR industry as it became.
Agreed. That's phenomenal that, it went from developing a methodology to screen to being part of a company and then being bought out. And
I didn't develop a methodology to screen. I developed it to understand something about the receptor, but I think I identified that it potentially could have utility in such a screen. And yes. I think, that's the important thing, maybe for many of your younger listeners here, do the work because you're asking an interesting scientific question, but just think if it might have applicability.
You make a great point there. And I think the science, the data, the science, and the scientific questions should be the driver. And then if that leads to a major discovery, or it leads to a technology that can be applied in order to discover new ligands or potential pharmaceuticals, then that's an added bonus.
Indeed. And, for example, one of the things I was intrigued by in the early years of having my own lab was we all know that treatment of cholera toxin results in a sustained elevation of cyclic AMP. But one of the things that actually perhaps people didn't realize at the time was that if you treat with cholera toxin and the GS protein becomes modified, it actually becomes downregulated very substantially. So rather than activating it permanently, with cholera, you get to a point where there's virtually no GS left in the cell. And it's an intriguing balance between synthesis and destruction of the protein.
How phenomenal. I asked this from everyone, and now getting to know you a little bit, the original question is, what is your favorite GPCR? But let's add in there, what is your favorite GPCR or potentially G protein and why?
Well, my favorite G protein has to be GO. Still, I think G other in terms of how it was first named, and simply because that was the purification of that was my first major publication. So it was a JBC paper going back to 1985, and it's still one of the very well cited papers from my career.
Wow. I'm thinking at 1985, I was 5 years old, which is...
Well, indeed. I wasn't much older, of course.
Of course. Of course.
But so that's definitely my favorite G protein because of that element of history. But when you talk about favorite receptors, because I came from a G protein background, certainly for the first 20 years or so of my career, I was very much a magpie. I simply picked up and used whichever receptor was going to be useful to allow me to turn on the G protein that I wanted to look at. But in recent years, I've become really fascinated by receptors for metabolites. And perhaps in the last 10 years in particular, my lab has been really well known for working on receptors for free fatty acids.
Yeah.
And a lot of the effort still in the group is either on those or on other really poorly characterized receptors that respond to components in our foodstuffs.
I think that's a very interesting topic because we need to better understand. So, they say you are what you eat. Yeah. And I strongly believe in that, because depending on what you eat, your body responds differently. And then it allows expression or repression of GPCRs or any other proteins that modulate your metabolism. How did you get into thinking about metabolism and the role of G proteins and G protein signaling, GPCR signaling in the metabolism?
Well, I think it became my focus based on a collaboration I had, and indeed still have, with a chemistry team based in Denmark. So a team led by Professor Trond Ulven, who's now at the University of Copenhagen. But at that time, when I first knew him, he was working for a small spin-out company and working on the receptor that's now called free fatty acid receptor 1, or in old money, GPR40. And it was very clear that this had big effects in regulating the release of insulin from pancreatic beta cells. And there was clearly an opportunity to maybe think about therapeutic strategies to enhance insulin secretion in a glucose-dependent manner. Now, when Trond left the company he was working with and went back into academia, he and I started to collaborate on a range of receptors for fatty acids, both for short-chain fatty acids, so FFA2 and FFA3, but also the long-chain fatty acid receptors, FFA4.
You made a couple of great points, and I'm trying to organize my thoughts because I have 3 or 4 questions coming into mind at the same time. You mentioned previously that you had this work with Arena Pharmaceuticals, and then you've been collaborating with this collaborator who had a biotech or pharma background. I understand based on our conversations that science guides and your scientific interests guide you in the choice of programs that you run. But it seems to me that you do have this inclination as well to use the technologies you develop to answer scientific questions, potentially in a more biotech environment. How do you reconcile those? Where did that interest come from, and how do you reconcile those? I think it's important for the younger generation to understand that when you're a professor, you follow the science, but you can spin out, or you can have multiple interests.
Yeah, and I think it was just a recognition that GPCRs are so important in drug discovery and in the way that therapeutic medicines have been developed, that perhaps in some ways I almost wanted, if you want, a piece of the action. But a good example would be that one of the areas where perhaps over the years I've been best known for making a contribution is in the idea that GPCRs don't always exist simply as monomers. Mm-hmm. So the idea of dimerization, which had been around for a little while, but particularly Michel Bouvier, who's at the University of Montreal, and I really started to push this concept at a technical level. Michel had really started to use resonance energy transfer-based approaches, and I pretty much piggybacked on some of his ideas to do some similar things. But again, in terms of technology, I developed another technology to allow people to screen for ligands that would selectively regulate dimeric forms of receptors. And again, I was able to persuade my university that we should patent the idea. Again, we then took that to a second American company that's actually on the east coast of the US this time. So I'm getting closer to the UK as we go through. And that company, Cara Therapeutics, we licensed the technology to, and Cara again now is a NASDAQ listed company.
Wow.
And I persuaded my university because the first time we had simply licensed a technology for cash, that we should take an equity stake in Cara as part of the process. And when Cara went public, of course, the university did rather well out of selling its shares in the now publicly traded company.
So scientist, biotechnology, thought leader, and business person. It's amazing.
And then subsequently, of course, in the last 5 or 6 years, I have founded 2 spin-out companies.
Amazing. Tell us a little bit more about that. How did that come together in your mind?
Well, the first one, which is called Caldan Therapeutics, which stands for Caledonia plus Denmark, so Scotland and Denmark, is based on my collaboration with the guy I mentioned earlier, Trond Ulvin. And it was really to exploit what we'd discovered and ligands we'd co-developed around these long-chain fatty acid receptors. So, sorry.
No, I was going to ask him, how about the second? The
second one? Well, the second one is kind of different because my main collaborator now in terms of science is a guy called Andrew Tobin, who I persuaded a few years ago to move from the University of Leicester to the University of Glasgow. Already we'd been working closely, and Celtic Pharma Therapeutics is Andrew and I's new company, but actually the main target is a malarial kinase and not a GPCR, although we do have GPCR programs as part of the company as well.
I'm excited to talk to Andrew in about 2 weeks.
Right. Well, he'll tell you more about that because the malarial kinase side of that is very much his baby. But he and I have worked now for more than 10 years together on various GPCRs, and we've really gone heavily away from what many groups have done in terms of moving into structural biology to use transgenic mouse lines to really understand the physiology of poorly characterized GPCRs.
I think that's also very important. And I've spoken to many guests, and some are very structural, some remain pharmacologists, and others go into animal models. And I think it's all of these 3 pillars, and there's many more others, are part of the same coin. It's important to know the structure, to understand the pharmacology, but also to understand the physiology of what happens.
Absolutely. And of course, we try to do all of these things, plus some others as well. But, you can only in the end perhaps be a master of a few things. And I think we made a conscious decision because there were so many strong groups heading very heavily into structure that we felt it was going to be very difficult to be truly international leading. And I think one of the things that many people would say about me is I'm not interested in being second at anything, whether in football or indeed in science. So we wanted to do something where we would be viewed as clearly the international leaders.
You made a fantastic point, and this goes back to the fact that you decided which way you wanted to go, is the fact that you looked out at what are you good at, what can you do, what you would like to do, and picked the thing that, had all those components. I'm very good at this, I can be the best at this, and I actually really enjoy doing this.
Yeah, absolutely. So that's a very good set of points to me. The other thing, which of course I'm quite keen on, is always pushing forward and learning new things. So one thing I'm definitely not content to do is sit on my laurels and not learn new things. So maybe 7 or 8 years ago, I started a project with a group of physicists at the University of Milwaukee, Wisconsin. And I think for about the first 5 years, we hardly understood a word each other was talking about. They were struggling to know what a cell was, and I had absolutely no idea about the mathematics that they did. But we've got a paper just coming out in PNAS together, which I think is a fantastic example of how you can combine these things. And often in PNAS, where they put the different areas of science, it's very unusual to see the two headlines as pharmacology and physics. And that will be a fun paper for people to look out for, which is looking at the dimeric structure of receptors actually in neurons of a transgenic mouse.
Wow, can't wait to see that one. And it will be linked to your podcast episode page. And
yeah, that would be very good. The other thing which I've been trying very hard to learn a little bit of, but I'm glad I've got a wonderful collaborator in the area. I think the way things are going now, beyond structure and static structures, are really taking advantage of molecular dynamic simulations and what we can do in terms of machine learning to understand how ligands bind to receptors and to develop these. So my good friend and collaborator, Irina Tikhanova, who's at the University of Queen's University, Belfast in Northern Ireland, is trying to help me to work through there. And intriguingly, of course, although she's a little bit younger than me, she was also a postdoc at NIH and had invited me to give a talk at NIH when I was going back to the US. And we started collaborating about 12 years ago now as well on aspects of structure and how we use computational chemistry to develop new ideas.
I think it's so wonderful to see the breadth and the depth of the work that you've been doing over the years. And going back to the point where you mentioned that you really wanted to find the niche in which you could be number one or internationally recognized for that. But in addition to that, I love the fact that you want to learn new things. And the best way to do it is to work with the experts at that and having collaborations.
Absolutely. But I think this is one of the joys of being in science generally.
Yeah.
Obviously, people have got quite distinct skills, but often are bringing different technologies and different insights into the same type of questions. And of course, collaboration is wonderful, particularly if you like the people you're collaborating with. So I think, if you're good friends with someone you collaborate with, it just makes the whole thing so much more enjoyable.
Absolutely. And I think
I would say that about all my key collaborators. I like them all very much as people as well as scientists.
You made yet another great point. I think the relationships that you developed are very important in order to facilitate that collaboration, because it's interesting because when I talk to non-scientists, they think that scientists typically work alone. They don't talk to anyone, and you're alone in the lab late at night with your lab coat and pipetting. And the reality is that science is a very collaborative effort, and it does help to really like your collaborators in order to actually have fun at the end of the day.
Yeah, well, I definitely believe science should be a very collaborative effort. I'm not sure it always is. Yeah. There are certain tensions sometimes about competitiveness and priority. And of course, there are challenges sometimes in the divide between academia, where mostly we are given our research funding from governments and charities and public agencies, where it's important that we make that information as widely available as we possibly can in open access and to make it readily available to people. But of course, sometimes you want to collaborate with people who are part of an industrial company or process, and they, of course, have to be slightly more careful about what they might say publicly. And I guess that's been a learning experience for me also in terms of moving into and having founded spin-out companies, because I'm one of these people, as soon as I get a new result, I want to tell somebody. But sometimes from a company perspective, that's obviously not really the sensible way to go forward.
Agreed. And again, you made a great point, and I don't think we talk about it enough, that you can only collaborate with the people who actually can, who want to collaborate. And yes, in science, there is competitiveness. There are egos as well. But I think beyond that, and I always try to focus on the data and on the science. But the first step is really to like that person that you want to work with. And it goes both ways as well.
Yeah. And of course, there are sometimes frustrations as a pharmacologist when you're able perhaps to access some tools or ligands from a commercial organization. And then, of course, it may be difficult to publish on those quite as quickly as you would like. There may be provisions set aside where at a certain time point, that's fine. But, that does create challenges, not insurmountable ones by any means, but, I'm very, very keen on making data available to people as quickly as possible. It's one of the reasons I quite like some of the preprint servers, and some of the information you can pick up from there is really very helpful indeed.
It is. But I think that's how the game is played when it comes to biotech and intellectual property, that there are sacrifices to be made. Then again, if you get to launch a company or work with a company, and in the end you're going to end up with a drug that can help people, then it may be worth it.
Oh no, of course, it's a balance.
It is.
And I guess the other thing that's worth saying, obviously I've been in this field for a very long time, but of course, as a university academic, a substantial part of my role is as an educator. So I'm delighted that, many people who've trained in my team have gone on either to run their own labs, or perhaps just as many have actually moved into the biopharmaceutical industry. And obviously that mix of pharmacology and drug design hopefully has stood them in good stead.
Again, absolutely great point, which took me to the question of how do you do it? Because from the outside, from the conversation that we've had and for everything that I've seen about you, it seems that, everything was, well organized, well thought out, and things happened, very smoothly. So here's two questions. One, how do you do it to, teach, be a professor at the university, run your own team, work in biotech? And also the second part of that question is, what were some of the difficulties that you had to overcome to get to where you are today?
Okay, so I guess in terms of the aspect of being organized, I guess just I am quite organized. I forward plan a lot. Mm-hmm. I think I'm quite perspicacious. So I quite like language as well, Yamina. So I do quite like to get big words into my papers. And then the reviewers tell me to take them out because it's not really an English novel I'm writing. In terms of challenges, I think like any academic, I've had quite a few grants turned down over the years. It's always frustrating when you get to a point where really that puts a significant delay on your capacity to move forward, because in many situations, certainly here in the UK, broadly, if you submit a grant and it is rejected, then it's 2 years before you're allowed to put a grant in on the same topic again.
Wow.
So, that can be quite a deleterious process in terms of trying to keep momentum in any one field. So maybe that's why I also have programs on quite a number of receptors, so that hopefully at least some of them are always moving forward, while others are having some teething problems in getting funding. The other challenges, I don't think there are many really. It's just a joy to be in a job where, as long as you're able to attract funding, you can pretty much ask the questions you want to ask. And I'm fortunate enough that my teaching commitments are not so all enveloping that I don't have time for research.
And it sounds to
Over the years, I've taken on a lot of university level commitments. So until last year, I was Dean for Research for the 11 years before that. And that was quite time consuming. But I learned a heck of a lot, certainly on topics that I knew virtually nothing about before that. And I found that also really quite enlightening in terms of the processes that universities use to run by.
Interesting. It sounds to me that you really had a lot of fun getting into new projects independently of the topic, whether it was university administrative functions, teaching, biotech, pharma collaborations, or scientific collaborations.
Yeah, and I still really enjoy teaching. So whether it's to relatively junior undergraduates or courses maybe for master's level students, I think, passing on some of your experience and some of your thought processes can be really a very endearing process.
Amazing. What is your advice to junior scientists who aspire to contribute to science, but also to the field?
Go for it. I think, have your dream and go for it. Now, I think the challenge, of course, is that many people who perhaps train to postdoctoral level, the reality is there aren't really the number of academic posts available to allow that to happen. But if you don't try, you'll never get there.
Yeah.
And the other thing is, I don't think I'm very good. I think I just work hard. So a sort of Presbyterian work ethic never goes to harm in these areas either.
But there's also the fact that you really enjoy what you're doing.
Oh, absolutely. If I didn't enjoy this, I would have probably certainly not put in the hours. I think it's been a struggle for me in the last few years, but I think many of my peers who are of a similar stage in the career to me would have said that I was one of the best informed and most knowledgeable people in terms of the field, in terms of reading. I must say I'm struggling to keep up with that as fully as I would like these days, but I think that's just the vast volume of information there is out there.
Well, it's so interesting that you mentioned that, and it is a compliment that actually I was at the GPCR, Great Lakes GPCR retreat about 2 weeks ago, and I was talking to Michel Bouvier, who said exactly what you just mentioned, that he not only he thinks very highly of you, but he is in awe at the fact that you know so much about the field and you're up to date and you know so many things. And I thought it was, and I actually told him that we were recording soon our podcast episode. So I'm glad that, I heard that from him, but I already knew that.
I'm glad we have a mutual appreciation society.
Yes.
Because, we've been in the field very much as peers for a very, very similar length of time. And I guess equally, there are many themes that we've both worked on. We haven't worked together very much, which is perhaps one of the disappointments over the years. But we've worked on very similar projects. And of course, we've seen each other at many events around the planet.
Well, this may be, the beginning of something. Maybe Michelle will listen to this podcast episode, and it might lead to a collaboration.
You never know. You never know.
Absolutely not. All right. Two last questions before I let you go today. We've talked extensively about the work that you've been doing and how you got from, purifying G proteins to GPCRs and developing all these tools to better understand the pharmacology, the biology, but also how you leverage that to take these tools into biotech pharma settings. What would be the top 3 aha moments that you had as a scientist and that shaped your trajectory?
Gosh, that's a big question, because I would like to say I've had lots of them. But the reality is, of course, many people don't have a great number of great ideas. So I think the first one for me was really the recognition, maybe driven a little bit by the work of Heidi Ham, that the C-terminal tail of the G protein, alpha subunit, which I guess in these days is the alpha 5 helix, and we all know about its detail, that was really going to be the integral part at defining how you could selectively interact receptors and G protein pairs. I guess the second was really the idea, which I worked on for many years, that the dimeric organization or the multimeric organization of GPCRs might be really quite important. And that was something that was worth pursuing. And the way that allowed biophysics to come into my work as at that time, basically a biochemist or a pharmacologist. And then finally, the recognition that really, yes, we could start to utilize these very, very poorly characterized receptors as drug targets, and to break away from just thinking that the beta-2 adrenoceptor was the only receptor that was worth studying. And many apologies to Bob.
I think it's so interesting. People who are not necessarily in the field but know of the GPCR field, every time they hear beta-adrenergic receptor, they think of Bob, and vice versa.
Well, I think it's well deserved, and obviously Brian Kobilka might have something to say about that also. But obviously Bob has absolutely been the father figure to the field in developing many of these thoughts and ideas.
Absolutely. And the beta-adrenergic receptor. Actually, I've been trying to get Brian Kobilka on the podcast. It hasn't happened yet, but it's definitely in my plans to hope and have him.
Well, I'm sure if you tell him Michelle did it, he'll want to do it.
Oh, that's a great point. I told him Bob did it, but okay. I think I'll try and push a little bit further than that. Okay, actually, I have 2 last questions. One, really simple. If you weren't a scientist, what would you be doing today?
If I wasn't a scientist, I would be milking cows, because that was the big career decision point in many ways. And to be honest, if my dad had owned the farm rather than just working on it, that's probably where I would have ended up, still being in farming.
Would you be happy as a farmer if you weren't a scientist?
I'd be happy, probably, because I wouldn't have known anything else.
All right, last question. If and when you have positions open in your lab, how can people find you?
They either find me, they mainly find me through the website and at the Centre for Translational Pharmacology at the University of Glasgow. We frequently have positions both in the academic side and obviously, as I was saying, the new company that Andrew Tobin and I co-lead, we have a number of positions available there also. So we're easy to find.
Amazing. Thank you so much, Graeme.
Okay, it's been a real pleasure to talk to you.
Thank you so much.
Okay, goodbye.
Bye-bye. Thank you for listening to this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest and Ines Pinheiro. Become a beta tester today and sign up for the new and improved Dr. GPCR Ecosystem 2.0 at ecosystem.drgpcr.com. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. As always, if you have any questions or suggestions, email us at hello@drgpcr.com. Until next time, stay safe.